Nowadays, cancer is a major health problem worldwide. From the past, research and efforts on developing cancer treatment has been on process and showed curation effect with therapies such as targeted cancer therapy. Despite the therapy's duration of clinical response is limited by inevitable development of drug resistance. Even with the development of medical technology, evolution of drug-resistant clone is still unknown. Lung cancer is the cancer with the highest mortality and incidence among all cancer cell lines according to Cancer Statistic 2021. Therefore, our team have planned to establish TKI-tolerant lung cancer cell line and to find its mechanism, which will eventually play a big role in further biological drugs. Before starting an actual experiment, we didn't have much information about drug tolerance, so we first collected information about it by reading lots of articles. We chose H9075 and PC9 as our lung cancer cell line and chose osimertinib and gefitinib as drugs since it's generally used for actual patients. We searched several articles to find the concentration of the drug where the drug-tolerant cell lines occur after treating osimertinib and gefitinib. As an example, IC50 value of osimertinib where H1975 cell line become drug tolerant is 0.01 micromole, whereas IC50 value of gefitinib where PC9 cell line becomes drug tolerant as 0.1 micromole per liter. Also, we affirmed time and process of generating drug tolerant cell line through background research. We were able to design and establish our experiment plan with these articles as a reference. In order to figure out IC50 value of H9075 and PC9 cell lines, we have to progress cell viability assay several times. Out of old assay method, our team decided to use cell titer glue luminescent cell viability assay, which is known to have the most efficiency. We plated the cell in multiple of 2 and optimized the most efficient amount of reagent for our experiment plan. Finally, we were able to choose 50 microliters as the optimized amount of the reagent. In order to generate cell line at drug tolerant state, we first have to figure out the IC50 value of the drug to the cell line. We did luminescent cell viability assay with H9075 and PC9 cell line with osimertinib and gefitinib. We plated 10,000 cells on 69 well plate and progressed the experiment by technical triplicate and biological duplicate. Our cells were treated with the drugs in multiple of 10 in the range from 0.001 to 10. PC9 cell line showed the same result as the previously researched background information. However, H9075 showed different results compared to the previously researched background information. As previously mentioned, IC50 value of H9075 to osimertinib is 0.01 micromole per liter. Our experiment didn't show IC50 value of 0.01 micromole, but at 10 micromole, the difference was 100-fold. We believe that this was the problem with H9075 cell line since the drugs worked well in PC9. Therefore, we were luckily able to receive H9075 cell lines from other agencies and re-experiment cell viability assay. However, these cells from other agencies also didn't show ideal IC50 value of osimertinib. Since drug tolerance occurs later than early resistance and the number of established drug-tolerant cell line is very small, our team first plated PC9 H9075 cell lines by 10,000 per 500 wells on 96 well plate, each member responsible for 6 plates. We treated the cells with the drug by IC50 value and established by previous procedure twice a week. This step is a long process. According to the journals we studied, about 3-6 months are required in order to generate cell line at drug tolerant state. In general, cells should shrink and eventually die under the drug treatment. But during the drug treatment on PC9 we could observe something unusual. First, we considered the unusual phenomena as cell colonies. But one time there was a blackout and there was a problem with the CO2 incubator, where we kept our cells in. Considering this situation, we infer there was a contamination on cells during this blackout. In case of H9075, as previously mentioned, our team was not able to get the ideal drug IC50 value. If it was in normal situation, H9075 should be dead with osimertinib treatment, but the result we obtained by performing cell viability assay showed H9075 having resistance toward osimertinib. We thought that this could be due to the problem with the drug or the cell line. In the case of the drug, we used the same drug to treat PC9 for the validation of IC50 value, and it showed ideal result, which proves that the undesired result has nothing to do with the drug. So we judged that there was a problem with our laboratory H9075 cell line. To solve this problem, our team searched for laboratories that owns H9075 cell lines. Our contact with them were successful, and we performed cell viability assay on H9075 several times. However, we couldn't achieve the ideal IC50 value. Since H9075 didn't show ideal IC50 value toward osimertinib, we decided to analyze whether the cell line obtains T790M mutation or not, since ideal H9075 cell contains this mutation. To do this, we performed genomic DNA preparation, and we are planning to identify the sequence by using the sequencing service. Therefore, currently our team is in the middle of the generating cell line at drug tolerance stage once again. Next, our team is studying about pros and cons of various CRISPR pool libraries, since we are planning a CRISPR library screening after generating drug-tolerant cell line. Our ultimate aim is to unveil the genes and pathways that are necessary in the establishment of drug resistance. Hence, we need to trace the cell to identify which type of cell acquired the drug resistance. In order to do so, we are also planning to barcode every single cell by utilizing a barcode library. In addition, since our aim is to identify pivotal genes and pathways of drug-resistant establishment, we also plan to perform RNA sequencing. We believe that this procedure will clarify the result of CRISPR library screening. Overall, during the last 4 months, our team researched plenty of articles related to drug-tolerant cell lines, such as cell viability assay, reagent amount optimization, drug IC50 value validation, drug-tolerant cell line generation, genomic DNA preparation. We also performed a variety of experiments, such as with cell viability assay, genomic DNA preparation, drug-tolerant cell line establishment, etc. We were able to optimize the quantity of the reagent used in cell viability assay, validate ideal IC50 value for both gefitinib and osimertinib on PC9 cell line. On the other hand, we had difficulty in validating IC50 value for osimertinib on H9075 cell line. We were not able to reach the final step of this project in time, since the process of generating drug-tolerant cell line takes 3 to 6 months. But we are looking forward to identifying necessary genes and pathway in drug tolerance through CRISPR library screening and RNA sequencing. Ultimately, we will identify the drug tolerance inhibition mechanism. In a nutshell, we strongly believe that establishment and further research of drug tolerant cell line will be able to contribute to anti-cancer therapy, especially on lung cancer patients who are resistant to lung cancer target therapy. If we are able to find the mechanism and inhibitor of drug tolerance, we truly believe that this will contribute to efficient biodrug industry. Thank you for your listening.